Yes. Good morning to all. Welcome to the fourth day of the international webinar series on infectious disease, biology, diagnosis, preventive strategies of coronaviruses. Let us invoke God's presence with a prayer. Loving Heavenly Father, as we gather together, we praise you for this day and your purpose for it. Bless our program, the speaker, the organizing team, and the participants as we sit in your presence today. Recalibrate our intentions and refocus our hearts. Give us renewed strength and courage to obey you. May your peace rain down on us today. This we ask in Lord's name. Once again, I welcome you all. I deem it a great honor to introduce our speaker for the day, Dr. Raghavan Palaniappan, Senior Scientist, Research and Development, IDEXX Laboratories, U.S.A. Dr. Raghavan completed his PhD from Bharathidasan University. He finished his post from CCMB. He has been the Research Associate at Cornell University, U.S.A. He is a highly accomplished senior level staff scientist with experiences in discovery of biomarkers, protein purification, immunoassay development, and therapeutic. He is an able research team leader with technical expertise in developing new diagnostic assays. A proactive and participative manager with strong analytical skills with a focus on innovation. He has received 5 patents for invention of vaccine and diagnostic reagents against leptospirosis. Dr. Raghavan is a reviewer of many international journals and also has numerous research publications in peer-reviewed journals with high impact factor and citations. Thank you for the acceptance to share your research knowledge, sir. Over to you.
This is Raghavan Palaniappan from Portland, Maine. Actually, it's you may all have to wish me good night here because it's already 12:30 here. So first of all, I would like to thank the patrons, the principal of Loyola College, and also the organizing committee for this conference, and my senior, Dr. Agastian, and also Jancy for providing, for giving my background about my microbiology background, and also about my experiences in biotechnology. I'm pretty excited to share my experience with you all. And so to start with, what I want to do is let me just go to the agenda first. And this is my agenda for talk today. What I want to do is I just split my talk into different fragmentations. I work on discovery to diagnosis right from my master's to my postdoc, and also even now. So since my senior, Dr. Agastian, has requested me that there are a lot of young people, students, and scholars will be in this webinar. So he told me that probably it would be good to give about how to improve the scientific skills, and also try to highlight some of the importance about the discovery and also for the diagnostic part. So what I did is I just initially, the first half of my presentation, I'm going to pull out some slides about the traditional method of what the cloning means, and also what is the modern approach that people are doing right now, and also what type of purification they are using it, and also about the immunoassays. And then I will go deeper into my favorite field of disease pathogenesis and prevention. I always like to start with acknowledgement because without these people, I wouldn't be the person who I am today. First of all, I would like to thank my master's mentor, Dr. Jeevanand, because my passion for scientific studies, it started right from this Virendra College, where I did my master's in microbiology. Um, when I was doing my master's, I did my thesis on studying Salmonella typhi and Typhimurium. So I had to go to the field to get the samples, isolate the organisms, and then once when you find out which is the organism is Salmonella typhi, then we did some animal model to see whether those Salmonella typhi and Typhimurium, which carries the multi-drug resistant plasmid, whether they could able to transfer those plasmid into the normal E. coli system, normal E. coli in our intestine. So that's what I started doing it. And the person who guided me, Mr. Jeevanand, his inspiration and also his dedication to research really impressed me a lot. So I learned so much from him. And then I went on to do my PhD in biotechnology. I have to thank CSIR and also UGC for providing me scholarship. When I moved to Dr. Vivekananda's lab, so I learned about the physiology and also the molecular aspects of Pseudomonas. And he gave me a lot of opportunities, and he actually shaped me. There I started working on isolating some extremophiles. And actually, Shivaji is a well-known expert in the field of microbiology. I learned, if anyone is, if people know about Bedge's Manual of Microbiology, you will have like a series of tests for detecting organisms. So I did most of the tests in that Berge's manual when I was in CCMB. And then I also learned a lot about sequencing and also remote typing. And so with all these skill set, I got offer from 3 different places. Those Professor Chang at Cornell University. The reason for choosing this university is the work is more of application, and because I thought maybe that one has more value, and so I picked that. Initially, I started working on leptospirosis. For the first few years, I started working on the basics of leptospirosis. Try to culture the organisms, and then so the overall goal for that project is to identify immunogenic protein. So I did library screening, and I did some cloning and also purifications. And what the important thing about at Cornell at Cornell University, I learned not only on the disease, I learned so much from this person, Professor Chang. He's a very dedicated person, and he's the first one to create animal model for Lyme disease. So he's an expert in, and also he's a very good genetics. Like he did a lot of work on mutations, variant population using in vivo and in vitro studies. So I had a good opportunity to work with him. And then for a while I worked, and then I moved on to foodborne disease, where I had opportunity to use the bioinformatic tools to do some comparative, and then identify some unique and specific genes. And then I developed a microarray for the diagnosis of E. coli and Salmonella. So that's a good learning curve for me. And then once when I land up onto IDEXX, here I had the opportunity. First, I was leading the project, and then I started working more on the protein purification side, where I was responsible for doing purification and also transferring the reagents to the pilot plant. Here I learned so much about protein purification. And then based on my skill set, I was selected to work more on developing a concepts for developing a test for the infectious disease. So then I worked on several issues here, and due to a lot of confidential issues, I cannot able to share some of the data that I have generated here. But I overview of what I did. And then finally, my parents, especially my dad and mom, and then I'm lucky to have a very good brothers and sisters, and my wife finally, because she's also a biochemist. She did her master's in. So first of all, I just want to give a kind of there will be more scholars and master students. I just want to go through the story of develop the skills. So this is just a slide. I usually go to a state science fair conference from high school though. So if you see some of the papers presented by these high school students are really amazing. I can just share a couple of posters with you. The one poster that I was really impressed is a girl, she had an equine farm, and she found one of her horses had erogenous infection, in the sense like it has an abscesses on the skin of the horse. What she did is, this is high project. So what she did is she isolated the organism, and then by just plating onto an agar plate, and then she took and she found somewhere in the internet that manuka honey, it's a kind of a honey that it has a lot of antimicrobial effect. And the manuka honey is quite famous here because originally I believe it came from New Zealand or Australia. So she honey and then study the inhibitory effect of that compound with the Pseudomonas aeruginosa. And she found there was some inhibition with that honey. And then later on, she took that and applied to that real horse. And then she picture, I was really amazed, day 1, day 2, day 3 pictures. And then how well that the honey helped those abscesses. I was really impressed with even though it was kind of small work, I was really impressed with the work. And then the second is this work, the one that I'm showing right here, studying the effect of microplastic on cell culture. So basically, if you see this, major abundant microplastics are polyethylene. The high school student, what she did is she had some collaboration with the University of New England, and she did a titration with in 2 different cell lines. One is human, one is I believe it is an MDCK cells and mouse fibroblast cells. And then she studied the cytotoxicity of these plastics, and then she also took that protein from those whole cell lysate from those cell culture. She ran it in a gel, and then she found there was a significant differences between the control and then polystyrene treated cells. And then she also did a qPCR where she did some oxidative stress marker and also cytokines, and then where she found there was an upregulation of cytokines and also the upregulation of superoxide dismutase. It was a pretty amazing. And then the student was awarded a top scholarship award in the sense she got a complete waiver for university. And the thing that I am impressed is it's not about science. It's all about working with people, and then most of the reagent, because the university can provide only space, right? So there, she had a very good people, 2 people. One is a professor there, and another one is a postdoc there. Both of them guided her throughout. It looks like the kid, they not only learn science, they learn how to negotiate with the different companies. So they reached out to different companies and they got the primers, got the reagents free from them. And then they also work as a kind of a team because it's working in the lab, like it's, you have to get input from others and then work towards the goal. And then it also helps them to be like independent when they grow up. So I was really impressed with, and so most of the posters I saw, like probably 250 to 300 posters. Most of them are high school posters. It was pretty amazing, and so they have a very good exposure compared to us, like especially like us. We study theoretical knowledge, but here they are studying more of a practical knowledge. That's what I found the differences, but it doesn't matter, right? So the passion, it comes either it comes from work that you did in school or the work that you did in master's. To me, it came when I was doing my master thesis. So when I landed onto Cornell, the first thing that my professor asked me to do is markers. As people who have done some lab work, and usually they will run a lambda DNA, and then I did some sucrose gradient, and then pulled out the DNA, and then that's the first thing that I tried. Each one in our lab have been tasked to do this laboratories, markers, and also we make our own homemade Taq polymerase. Especially at these circumstances, like COVID-19, where the PCR is used for diagnostic purpose, and so the Taq polymerase and these are really valuable. And so what we can do is back in India, like we can get those plasmid, by writing to the people, they can provide the plasmid free, and then even in the master's level or in the scholar level, they can start purifying, the Taq polymerase or marker, or they can be considered as a kind of project for them. These are all small things. So I just right here, let me pull out my laser pointer so I can show you. So this is the website. You can just see, like, you can, if you go to this website, they will just tell you how to purify the Taq polymerase. And you can also get the plasmid from, from this website. Other aspect is, since the bioinformatics is going to be it's going to be a good tool later on because genomic sequence have been sequenced. So we have reached in a stage where, you have a voluminous amount of data, and you can just play around with this, and you can build your own homology modeling with your protein. So you can do all sorts of things just right online, where you put the tertiary structure of the protein, and you can also see how the genome, how you can compare one genome to other genome, and then see how homology it is, and the tree map. So those are the things that you can do with these kind of tools. So now I'm switching gear. What I want to show here is, I think the slide, it clearly illustrates that at this going on, and so the pathogenesis is always challenging. So it's challenging developing a right appropriate diagnostic tool, and also a right treatment to kill the disease. Because new disease or new treatment plan, it's going to come all the time. But it's important for us to understand the disease, how it caused, and what makes that virus to change. So these are all very, and why it affects people, why it is not affecting certain set of people, why it is asymptomatic, why it is symptomatic. So we need to figure out this. And if it is asymptomatic, have an antibody in the system. So these are the things that we need to understand in order to develop a very good diagnostic tool. I have an example later on in my slide deck. So then I'm switching gear to see, this is overall pipeline for developing marker. You can see right from identifying the target till developing a proof of concept. So when I say diagnostic here, I'm just focusing, because diagnosis is kind of a broad term. What I'm focusing here is more about the immunoassays. which is like antigen test or an antibody test. So it's not PCR or other things. It's just about immunoassay. So because that's our company is doing, so I just want to give you an overview of that. So for any disease, it is really important to understand the disease first. How it caused. What happens, why certain population is get affected, and then why it is symptomatic, why it is yes, you need to understand that. And then next is like once when you understand about the disease, then we have to employ some kind of a techniques, like what kind of technique. So if the pathogen genome have been sequenced, then it is easier. So you can go and compare with the genome, you can pull out some membrane proteins or you can pull out the proteins, and then you can clone, express and purify those recombinant protein. And then you can start finding out how good it is an assay or for an antibody assay. If there is no genome sequence available, then we have to rely on some traditional methods like building genomic libraries and screening libraries, or we can go for some proteomics work. So those are the things that we can do. So I just put some here. You can see here some of the traditional methods, and then you can also go for high sequencing. The high throughput screening is like once when you know the genomic sequences, you will have, like what are those, because we always target any for vaccines or for diagnostic, we always target a membrane protein or secretory protein. These are the primary proteins that interacts with the host first. So if we target a diagnostic reagent or a vaccine against the proteins, then it would be you can prevent those infections. So we can do a high throughput sequencing, and then we can also comparative genomics, and we can do some proteomics work either by using a mass spec, or you can run 2-dimensional electrophoresis, and then you can also microarray. So these are all like different techniques that you can do. And you can Google it, you will get tons of data about this. What I'm going to do is, while doing this discovery to diagnostic, the 3 key points is how to rapidly develop a test, right? If you have been assigned, if you have been assigned to do some test for a disease, the first thing that you have to do is to understand the disease, right? Then later on, you have to go and see how we can clone this to make a protein. So what I'm going to show you here in the couple of slides, how rapidly you can clone the gene, and how well you can purify the recombinant protein, and then how well you can evaluate those antigen in an assay. So that's what I'm going to show. So the first one, what we always do is, so there are different expression system. So one is an E. coli expression system, and another one is a mammalian expression system. And there are also other type of expression like baculovirus. You can put it in Bacillus. So there are several systems. But the one that is used is E. coli and mammalian expression system. If you look into this, let me pull my pointer here. So the gene construct, you can order the gene construct. You can do the small scale expression, and then to a large scale expression. And then you can prove, you can get the protein, and then you can purify the protein, and then you can freeze thaw and study the stability of the protein. If you look into this window, the flow chart is quite, except here, you can see here, right here, right? So the differences is mammalian cells usually takes a lot of time, and we are living in a world like where we need to make the test rapid. So what we always do is we use E. coli primary thing, that where you can create the reagents very quickly, and then you can evaluate it within a week, right? So we rely on E. coli system initially, but there are some proteins where you have to express in mammalian version. So there is no way at all, because they may be a glycoproteins, because E. coli proteins, they won't glycosylate the protein. So in mammalian version, it's easy to produce a glycosylated protein because they have a very good post-translation modification system available. So let me pull this from 1D. Okay, so why E. coli? First of all, why people are widely using E. coli as a kind of a right? So the main thing is E. coli. If you look into this picture, it's a periplasmic space, So that's where they store those recombinant protein, right? So it's kind of a balance. So you can induce the gene of interest for a membrane protein. You can induce using IPDG, so that will just secrete a lot of proteins. But when you push the limit where it explodes, it will form inclusion bodies, right? So the inclusion bodies are just kind of a small vesicles where the recombinant protein gets folded in different form. In the past, like 10 years ago, 15 years ago, people, they will take the inclusion bodies, they will just treat with some denaturing reagents, and then they will purify the protein. But now there are different systems available, different promoters, different types of expression system available, different types of vectors available. So we can make the protein as soluble or recombinant. So I'm just throwing a couple of slides here, like just to show you what are the advantages of this E. coli expression system. So the best thing, as I pointed out earlier, it's a well-established system, right? Easy to manipulate, and the genes have been well studied, and it is very safe. And you can just grow it in a lab. It is very inexpensive. You just need a luria broth or just a normal broth. You can just throw the E. coli cell, and it will grow very fast, right? And then doing transformation is cheaper. You can just do the transformation very quickly. So it's easy to do, and it has some disadvantages, right? As I pointed out before, it lacks those post-translation modifications. So if you are trying to work on some glycoproteins like viral glycoproteins, you cannot use E. coli to express the recombinant protein. And then the solubility is another big challenge. You can resolve the by taking that E. coli recombinant protein and then putting into different buffer systems. And also the chaperones, that's another disadvantages. And I have already told you about the inclusion bodies and aggregation. So these are the things, like when you push the bacteria to a limit, like above the limit, it forms the inclusion body, and it also forms an aggregations. So how do we get, what is the most important thing, right? So the important thing is, getting a soluble and well-behaved recombinant protein is really crucial application. And it's either it's a vaccine or it's for a diagnostic reagent. So you can take the foreign plasmid, and here is the recombinant plasmid, and then you can transfer that into an E. coli cells. And then if E. coli cells protein, and it goes to the periplasm, and then you have the protein. But if you push the E. coli cells to a limit, like, if you want proteins, then it started forming inclusion bodies in the cytoplasm. So when you break open the cells, you will see some inclusion bodies, but the protein, the soluble protein is the important one, because that's where, the proteins are well folded, and it's easy, and then, you can use it for further use. In the past, like, people have been using widely about the inclusion bodies, like, they can take that inclusion body with, by treating with a denaturing urea, and then gonidium, and so then they refold it back, and then they check, they check that secondary structures, and then see, okay, it looks good, and then they started using it. But in the industrial sectors, like, it's very important to have like a soluble protein, because we'll be shipping that protein. It has to be aggregate, and it should not form multimeric. So it's very important to have like a soluble and well-behaved protein. That's a very important step for that thing. The second thing is, so I talk about the expression levels. So the technology has advanced within the last 5-10 years. When I started working at we used to do, even at CCMB, we used to do a lot of cloning. So when we do cloning, like, we didn't know that some proteins, they won't produce in a huge amount. So you will end up only with the low level of expression. So consider this one as an E. coli protein, and here is the plasmid. And then you can see that there is a very low level of expression here, right? So the technology has advanced. And then now what happened is you people started synthesizing it. So when they synthesize the genes, they have an algorithm, right? So in that algorithm, if you are taking a gene from a parasite trying to express in an E. coli cells, the parasites has a different codon. So the codons are those codons are not used by the E. coli cells. So the E. coli cells, they cannot able to express that protein in a huge amount. So what this does, it changes those codons. But the amino acid is going to be the same, but it changes those codons. And once in a way that it recognizes by the E. coli very well. So whatever codons that E. coli widely used have been used, in this algorithm. So when they does that, they synthesize based on those algorithm, and then once when they clone that same gene into codon optimized version of that, and when they clone into a E. coli cells, they can produce high level of protein. So this is widely used, and we use widely in the industries here. And then the next step is I'm just trying to compare the traditional approach and also the modern approach, how things have changed, right? So in the traditional approach, when 20 years ago, when I started working on this cloning, we always clone the PCR by, you do the gene, you do amplify by PCR and you put it into a plasmid, and then you transform. And then if you want to express into a different vectors, you can subclone into right? And then you express the protein. Now the technology, it takes a lot of time actually. It takes at least 7 to 10 days. Now it's all changed. Traditional approach, it would take, if you want to clone 4 to 5 different genes in different plasmids sometimes. And but the modern thing, you can use homology cloning, or I showed you about the overhang cohesive end cloning. By doing these kind of techniques, and also you use codon optimized genes, and so when you do this, and your rate of cloning increases, and the success rate is also increases. So whatever that we thought, in the sequencing of a gene nowadays, it's not tedious. I know it's quite easy nowadays because the technology is so fascinating and it's improving every day. And the second thing about is, whenever we try to clone any genes, in the past, we'll be just using one plasmid or two plasmid. And, sometimes you end up with good expression. Sometimes, you won't get with the good recombinant protein. But there are things that you can do now is combinatorial approach. You can take a gene and put into multiple plasmids. These are all expression plasmids. Each plasmid will have different tags. You will have different promoters. At the same time, you can also use different host cells, BL21D3 is the commonly used host cells. And you can also use the cells called as suffle cells. By using the suffle cells, the second is, the disulfide bond formation will be improved. So you get well-folded protein. And nowadays, NEB is selling cells where, you can push the E. coli cells to a lower temperature where you can able to get a good expression valuable recombinant protein. By doing this, though, you save tons of time. Look into the traditional approach and also the combinatorial approach. Combinatorial approach, you are studying, you are taking one gene and then studying in several different vectors at the same time, and then able to get an efficient recombinant protein. So we have to be very thankful to this kind of technology that's going on. And then this is just give you an overview of this. So you can play around with the different types of promoters. When there are different types of promoter in order to express a gene, the widely used is T7, T5. You can use tag promoters, and these are all different promoters. And you can also use different kind of tags for the recombinant protein. So here is where you clone the recombinant protein. Here is just the plasmid representation. And here is that markers, either it's an ampicillin resistant or kanamycin resistant. And then you right here, and then the next is immediately is your ribosome binding sites. And then these are all tags. You can put the tags either to the C terminal of your gene or to the amino terminal of gene, and then you can express and purify the recombinant protein. So it depends on how you want to express it, right? So for some toxic proteins, you cannot able to use the T7 promoters because it will kill the E. coli cells. You have to choose kind of a moderate promoter like a TAC promoters. So it's your hand, like it's up to you to choose what makes express the protein quite efficiently. Same way in the tag, most widely used is a his tag, and there are certain toxic proteins where it needs to be expressed along with these kind of a bigger tag like a maltose binding protein or GST. So for expressing them. I have the opportunity to work on almost all the tags in our company. And so by doing so, you can save a ton of time. And you can clearly see in this picture, it takes this much time in using the traditional approach. Now, it's codon optimized, send a gene to a company, GeneArt, within 5 days, they will put into a vector, whatever vector that you are saying, it will be there within 5 days. You will have that plasmid in your hand, and then you can just transform it to an E. coli cells and start working for the recombinant protein. So what I said is like the technology is improving rapidly, and we are just taking advantage of it to improve the process quite efficiently. So then the next part is recombinant protein. In the recombinant protein, so whenever any project is assigned to me, I look into the literature first, right? And then see what are the targets available. And if there are any target for particular diseases available, is that someone expressed the protein, or is the literature they have mentioned about it? And then if you identify a target, then I usually see like what is its close homology with the and then sometimes we do a lot of truncated version of that particular gene, and then try to express the protein. And then once when you find out what target you want to finally use it, and then the host is a very important thing. You can go to an E. coli, you can cells, or you can go to yeast, or you can go to the baculovirus. The simplest thing, like if you want to try quickly, is an E. coli cell, as I said earlier. And then you can also go to the mammalian cells. So this is very important for a process map. And then you can synthesize the gene, and then you can study the small scale expression, and then you can go and do a large scale expression. This is an overall protein expression workflow for any project that I always do. So just to give you an idea of what is a typical purification method, right? So now to the next part, which is more about the purification. So earlier I talked about the cloning, and now I'm just talking about purifications. So in the purification, you have different set of chromatography you can use. IMAC purification. You can use ion exchange, which we all, even in the master's level, people have done some ion exchange purification. And you can also use HIC, hydrophobic interactive chromatography. You can do SCC, so many things like you can do with the pure. But the simplest way, I'm just trying to tell you about the simplest way of purifying the protein. The protein has tag, especially his tag. The his tag thing is, see whether when you break open the E. coli cells, see whether that particular protein is in a soluble form, in the sense like the buffer that you are choosing is really important. And so when you choose appropriate buffer, and if the protein looks soluble, and then you are taking those soluble protein and then putting back into an IMAC column. IMAC is a histratag, the protein of your interest, which has a histidine at the end, gets trapped in those columns, and then all others are flushed through. And then still you may get a lot of E. coli protein after those purification. And then you dialyze it. so dialyze in the sense you will be removing imidazoles that you are using to elute this protein out. And then after dialysis, you go back to do the ion exchange, you do the dialysis. So this is kind of a general method we always do in our lab. And the machine here is this machine is called as Acta-Avant. So fascinating. We can run several purification methods in this. And in the past, we used to have like a machine called as Explorer Purifier like that. This is really a Windows-driven. This machine is, you can have like a 10-liter culture. You can just purify with this machine with a higher flow rate. Not only that, you can just, they're all Windows-driven program, so you can just insert your E. coli lysate into this, and then you can just drive the program. You can ask to put it back into an IMAC column or your ion exchange column. It will be automatically processed, and then by the end, it get collected at 4 degrees. So they have their own 4 degree within this. So this makes the process quite easy, and it is also controlled remotely. So you can control from home. So you can do also here. For master students, this may be kind of too much, but what you can understand here is there are some process simple process. It's available to purify the recombinant protein. So just to show you an example of this, here is, so I'm taking a protein. So E. coli is expressing a protein. So the first thing that we always do is we will look into value of the protein. What is the theoretical pH value of the protein? In what buffer the protein is really happy? Is it looking happy in a pH, or is it looking happy in a higher pH or in a lower pH? So we'll try to figure out in what buffer the protein is happy. If it is happy in the sense if it is in a soluble form. So when you take the E. coli cells, you lyse the E. coli cells with the appropriate, and then you just run it in a gel and see whether you are seeing that protein in a huge amount. So here is the SGS page gel that here is the marker, the first lane, and then you can see this is the lysate. And then lysate is you take just use French press to lyse the cells, and then after that you centrifuge it, and then take the supernatant where you have the tons of recombinant protein that I'm pointing out is right here. I believe you could be able to see it with my laser right here. And you can see how good it is, neutral pH. And it is also looking okay with this carbonate buffer. And then when we figure out, okay, the protein is, most of the component, the most of the protein, it gets solubilized in the pH 7, then we take this material and put it back into a column called as IMAC. IMAC is just the histograph column. So these proteins will have at the C terminal or at the N terminal end will have that histidine. This histidine will go and bind onto the IMAC column, and then where you can purify the protein. And here is just a typical, you can see that purification. The lane, this is the lysate, and you can see this is the unbound form. And you can see clearly that most of the protein right here, it gets bind here. And when you elute the protein with the chelating agents, you can see this band, this peak, you can see here all these proteins. But even after this, you will see a lot of contaminants here. So in order to remove this contaminant, then we have to find out what is the pH range of that particular protein. And then we chose here an anion exchange column. You can see here we use, I use SP column. So when here, we could be able to clearly remove those contaminants that we are seeing right on this bottom into the next chromatography. You can clearly see it comes out very well here. And it doesn't stop right here. Once when you have the protein at the end, then we will look for this and then see whether the protein precipitate after storage. So we'll just freeze the protein and thaw the protein and just see it looks. And we do also a lot of analytical tools, just running SEC to see whether it is forming any kind of a multimeric form. And we also check the immunogenicity of the protein. Sometimes we look into the secondary structure of the protein. And so those are the essential analytical tools use before going back and check the functional properties of the protein. So I'm switching gear here. So the next aspect is immunoassay. Immunoassay is basically there are different types. What we always use is an antibody assay and an antigen assay. So what is an antibody assay? Antibody assay is detecting antibodies in the serum, right? If a pathogen enters human or animals, so by means of that, indirectly you can detect, okay, that particular patient is infected with a virus or a bacterial infection. So then what is an antigen assay? The antigen assay is very important because in the early stage, because antibody usually comes after day 7, you will get IgM a little bit, and then after that you get the IgG antibodies. On early part of any infection, antigen assay is very crucial, right? And it always correlates well with the PCR work. The problem with the PCR assay for any diagnostic purpose is if the organ or your immune system is good enough, or if they are treated, or if they had the infection in the past, and if the pathogen is dead, you cannot be able to differentiate by PCR. But however, the antigen test is always very important. It helps us to provide early diagnostic. So we have a test mostly with antigen test, especially for some disease. For some disease, we'll work for an antibody assays. So I'm going to just give you an overview about 3 different assays. This is straightforward. Anybody would see this, and you take the plate. You code the antigens, and if you have a serum sample, and if you have infected antibodies, the infected antibodies will go and bind onto the recombinant protein, and then you treat, you add anti-IgG, and then you can conjugate whether it is a peroxide or whether it is a HERPO or whether it is alkaline phosphatase. You can detect that by ELISA assay, right? So this is straightforward. Most of the people will be using. And the next one is a direct assay, which we use quite often in our lab. The importance of the direct sandwich assay, where you take the recombinant protein, you code onto the plates, and the same recombinant protein, you biotinylate the recombinant protein, and then mix both of them together in a serum sample. So your antibody, which is present in the blood, it gets sandwiched between 2 different antigens, and then it's easy to detect. So what is the advantage of this, right? So the main advantage is detect sometime IgM and also the IgG here. So it gets trapped right between the assay, right? And this is a format that widely used even for the lateral flow studies. And similarly, right here, you are trapping this antibody here. You can trap antigen here, right? You can you develop a polyclonal antibody or monoclonal antibody at some targets, and you can just code onto the plates, and then you can label the same monoclonal antibody or polyclonal antibody, and then just add those antibody to the samples. And if there is any antigen available, it gets trapped here. So this is trapping the antigen here. This is an antibody capture assay, and this is an antigen capture assay. Okay, so this is another kind of assay. And then the third part is a competitive ELISA assay. So what, right? So if I have a monoclonal antibody, for example, against any virus, what I will do is that particular monoclonal antibodies should what we call as sometimes we call it as an infected antibody. So it should match with the infected sample. So if with that particular monoclonal antibody, if it goes to some immunodominant epitope of that particular antigen, it will go and block that particular epitope. So if the serum sample, if they have infected serum samples, it won't go and bind onto this, right? So it means that the color will become colorless. So I'm just, for the comparison's sake, I put it right here on this side. So in that way, the antibody competes with its serum sample, and it blocks it. So you won't get color at all. So this is kind of another assay that we do all the time in our lab. So the next aspect is I'm just going deeper now into two specific disease. One is leptospirosis, which I did this. And then the next one is how to use the bioinformatic tool. So these are the two different things that I want to show you. The first is the So for leptospirosis, we use some traditional method. Remember, I was talking about the traditional method. Just to give you an overview about the leptospirosis, it's a kind of zoonotic disease. And there are different bacterium called as Leptospira interrogans. And there are several different types of species or serovars. And it is present in both animals and humans. And you may get infection from animals either by direct or indirect contact. And then the symptoms is usually you will have a high fever. Sometimes it also have the similar symptoms like jaundice. It may cause renal failure depending upon the serovar. Sometimes it may even cause multi-organ failure. Do we have any diagnostic tool for the disease? Yes, we do have that. But the diagnostic, especially it's not highly sensitive. What we do is called as a microsilation test assay. And also sometimes we do PCR, and sometimes in the clinical chemistry results, like doing a blood chemistry, looking into the white blood cells and also looking into the blood. Sometimes it is helpful. But is there any vaccine for this disease? Yes, they use whole cell vaccine for the disease, but it is widely available only in animals and not in humans. And find out early, it is treatable, and it can be treatable with normal antibiotics like doxycycline. You can treat it. So this is overall view about the leptospirosis. So here is a picture that I just want to show you to see how serious that particular disease. It's mostly present in warm place, tropical countries, and you can see that some of the rodents, symptomatic carriers, they don't cause any disease in these animals, but it will keep transferring that to animals and also to humans. It's present in most of the animals, like you can see dogs, pigs, and also humans. So it is, it affects almost all the organisms. And depending upon the type of serovar, it also multi-organ failure. Here is some of the diagnostic that I was talking about. So the microscopic agglutination test is widely available, but the problem is it's highly laborious, right? The laborious in the sense, even culturing the leptospira, it will take 7 to 10 days. And then once you use your serum sample and see whether particular concentration of the serum sample, you dilute it and then see whether it causes any agglutination. So this is really a cumbersome process. And then the next step is a PCR. That's quite readily available. But the problem with the PCR is sometimes, especially in animals, it's very difficult because vaccinated with the whole cell vaccine, then it's very difficult to figure out in PCR. And moreover, it presents in the blood only for first 4 or 5 days. And then later on, after that, it will go and settle in kidney or in liver, depending upon the serovar. And then the next is immunohistochemistry. That's also, and the commercial tests like lateral flow assays, they're all available. But the main important diagnostic is like, whenever there is an infection, but if you could able to the earliest stage, trying to find out whether the antigen is present in the blood, that kind of test is not at all available. So I'm just trying to find, I'm just trying to give you an overview about the protein that I identified. This is protein called as leptospira immunoglobulin-like protein. And this protein I identified by screening library. This protein is very important for leptospirosis. because this is a protein that primarily interact with the host cells, and we test using this protein, and we also develop a vaccine with this protein, and so we have several patents with this protein. And what is the peculiar thing about this protein? So the peculiar thing about this protein, what you are seeing is 1, 2, 3, 4. This one is a 90 amino acid repeats. So this is we called as a Lig-A because it is a leptospiral immunoglobulin-like protein because globulin. So in the database, if you go to the PFAM family, you will see a separate family called as immunoglobulin-like protein. This nature of this, based on the nature of this protein, it falls into that particular family. So the 1, 2, 3, 4, 5, 6, it all on the repeats. This is each one is a 90 amino acid repeat that goes 12 or 13 times. In the other thing is this is another protein called as Lig-B. It has the same feature, and then you can see the color. This overall, what I labeled as a Lig-A region. This is almost like 90% homology in this region between A and B, and the differences is a huge actually in the C-terminal region. It's only 20 to 30% homology in the C-terminal region. So how did I identify this protein? So what I did, I identified this protein by screening we took the leptospirosis, isolate the genomic DNA, and we built the genomic library, and then we screened the libraries, and we got several in the library, and then by using Bioinformatics, we found this protein is a membrane, it's a secretory protein. And then once when we identify that as a secretory protein, we express the protein, and then we prove that it could be a good diagnostic reagent for detecting the levels. It may look very simple here, but it's a lot of work actually. So the problem when I started working on this, the researchers who have been working on leptospirosis, they have been screening these libraries with the whole cell, the polyclonal antibody. So whenever they isolate the pathogens, they immunize into the rabbit, and then they generated the polyclonal antibodies, and they use these polyclonal antibodies to screen the libraries. So when we on this, we figure out that's not the right way to do it. We thought maybe we have to use a naturally infected sample. So we were looking for naturally infected samples, and we got one sample where the leptospira was isolated from that particular animal, and then we found that it is severely infected. Even the kidney was reduced, the kidney size was reduced in that particular animal. We took the serum sample from that particular animal, and then we screened the library. This is very important because you will see in a couple of slides why that naturally infected serum sample makes a significant difference in identifying this protein. So once when you identify the protein is really huge, it's initially we started working more on the protein called as Lig-A, which is the first part of the protein. Once when we identify, we express the protein as a recombinant protein. I couldn't able to express the complete protein, so I made pieces of it, and then we developed the polyclonal antibodies in rabbit. So we have the polyclonal antibodies in our hand. And then when I blot, so while doing the blot, what I did is here is the control, like E. coli with the Lig-A gene, and E. coli with Lig-A gene, the lane 2. And then third is we extracted the leptospira protein and then ran it in a gel. And then when we did the, you can see that the E. coli with the Lig-A protein, it only show up here, right? And we didn't see any band at all. So this was like, 20 years ago, I did this. At that time, so as a researcher, I was really frustrated with this. Why? Because we got the protein from the library, we had the polyclonal antibodies, and then why it's not lighting up? So that is something that it is going wrong into the literature. And then I found there are certain protein in the organism, leptospira, it gets upregulated or downregulated during infections. So then what we did, because leptospirosis usually it grows at 30 degrees centigrade. So I just higher temperature, 37 degrees centigrade. And then I used appropriate controls from our collaborators. We got the polyclonal antibodies from this. So this is one kind of protein called as L32. You can see here, this is a control that is grown at 30 degrees centigrade. And then this is 37 degree grown leptospirosis from day 2, day 3, day 4, day 5, and day 6. And you can see that it consistently, that particular protein expressed, right? Too. Same way, this protein is well known to be like a down regulator. When the temperature is raised, this protein will down regulate it. And you can see that it gets down regulated. And then looked into our protein, we didn't see anything at all. So, it was quite challenging at that time, because most of the papers, whenever they publish it, they will see the expression of the protein in their own organism. So, you purify protein of that particular target and then immunize into the rabbit, and then you have the polyclonal, and then when you screen back into the leptospira, and then if you are not seeing it, that's kind of frustrating, right? So then we thought, okay, how about this? Like maybe we should go and do electron microscopy. Maybe like immunogold labeling is a highly sensitive method. Maybe we have a chance to detect the leptospirosis. So we did gold labeling, but we didn't see any expression at all. So once again, there is no expression with the leak protein. Then we discussion about this, and then my professor told me maybe what we should do is we infect the hamsters with the leptospirosis, and then we take the kidney from the hamsters, use the antibody against the Lig-A protein to see whether that infected kidney have those proteins expressed. And then when we did that, we found the expression of that particular protein. And here on your top left, you can write here, top left, you can see expression. This is a kidney tissues, like, where I did the immunofluorescence assay. See those darker color, fluorescent color, it's expression of Lig-A. And then we use the whole leptospiral protein, we extract it and then put it into a rabbit, and we have the antibody against this as a kind of a positive control. And then same way, use another positive control here, and then a negative control is the pre-immunosols of this. So overall, what we found is it is expressing in vivo, but it is not expressing in vitro, right? And then what we found next is, okay, how about this? Let's see whether it has a value diagnostically. It's very difficult to express the complete proteins. So what I truncated this and labeled this as a conserved region of leak protein. And then I labeled this part as a variable region of A protein, then this part as a variable region of B protein. And then I expressed all these protein, and then I checked pure protein, and then I immediately checked the diagnostic value of this protein. And you can clearly see this. This is the MAT titer. You can see here that I took 26 samples, and then 23 of them were looking good in this titer value. And then once again, if you see here 11 samples from this 6400, we picked up 9. And then you will be surprised, why not 26 out of 26? Why not 11 out of 9, right? And we use serum samples. And the problem with the canine serum sample is the canine, usually they get vaccinated every year with whole cell protein. So the whole cell protein, whole cell vaccine. So they will always have an antibody against that whole cell protein. It always drives the titer value. So that's the big problem with the canines, because in dogs, it's very difficult between vaccination and infection. So in humans, there is no vaccine, so it's easy to detect. And then we also found, and also our collaborators, they found the utility of this protein. And then later on, we also use this protein as a vaccine. And this is one of the best vaccines for this disease. And it gives 90 to 100% immunoprotection against leptospirosis. And that's a pretty good protective efficacy for this vaccine. Overall, this is why Lig-A is a strong candidate for diagnostic. I just summarize it here because it is a surface exposed protein. as I pointed out, this protein is not present in high passage cultures. One of our collaborators, he took the leptospirosis and then put it into an hamsters, and then he repeatedly passaged the leptospirosis in hamsters. And then once when he get that, what we called as a low passage leptospirosis, these low passage leptospirosis, they express the protein. But making it, they are all made with the high passage leptospirosis. So they don't have that leak protein. So we found is the high passage protein, which people have been using it for vaccines. They don't have the leak protein, but leak protein is the one which gives a protective efficacy against leptosis. That's a great findings that we did. I think I'm running out of time to push within 15 minutes the next section. So the next aspect is differentiating E. coli pathotype. The bioinformatics is a fast moving field, and it's an integral part of biotechnology and computer science. And I think in future there will be more people working on the bioinformatics, considering the voluminous amount of genomic sequences. People will be spending more time on the laptop. They try to figure out how the protein interacts, how the protein how the protein can be used as a vaccine. So there are several tools available, and I'm pretty sure it's going to be widely employed in biotechnological industries. It has been using it already, but so 15 years ago we did this work. At that time, we thought maybe how we can use this microarray for detecting bacteria. So what we did is at that time the foodborne disease is widely prevalent. So we started, maybe we should start working on some of the E. coli and also Salmonella, which causes a lot of foodborne disease here. And so this is just a overview of how we did this. And you can see this is the plate, 96 well plates, where you can take the gene, and then you can pick some specific region based on the algorithm. And then that particular specific gene, it won't have any homology with other E. coli or other Salmonella. So you have to find out really a sweet spot for using it as a diagnostic reagent. So we found several genes like that, and then synthesize those 70 nucleotides, and then we put it into a slide of, we call this one as a microscopic slide. And then you can take, as a control, we took the real E. coli genomic DNA, and then we denatured them, and then we use random primers, and then radio label. Here I did the fluorescent label, fluorescent label probes, and then these probes were hybridized with those 70 MERS, 70 oligonucleotides, and then we use the GenePick software using the laser to find out which one is lighting up. This is very simple technique that we used, but the interesting thing is we could able to identify specimens within those E. coli pathotypes, and then try to figure out what genes we can use for the diagnosis. Now the technology has improved. I believe it or not, I can do it within 1 to 2 hours to do those comparative charts. So that's where the technology. So just to give you an overview of this thing, E. coli in general, they are non-pathogenic. They are called as opportunistic pathogens. They acquired those pathogenic virulence factor from other bacterium like Shigella or Salmonella or some other organ, either by transduction or by conjugation. It becomes a virulent organisms. And so here is I saw this in a Nature Review of Microbiology, where you can see that normal commensal E. coli, they have those virulence factor through the plasmid or through the bacteriophage or through the pathogenic island, and then they tend like a pathogens. By doing this, the E. coli converted into an enterotoxigenic E. coli, enterohemorrhagic E. coli, entero E. coli, uropathogenic E. coli, and meningitis E. coli. So there are different types of E. coli available. So our goal was at that time to see how we can identify those pathotypes by just microarray. So I did the comparative genomics of all the E. coli available at that time from the NCBA database, and then identify a set of parameters called as a common gene, specific genes, and then virulence genes, and then as a non-pathogenic specific gene as a kind of control. And then we did the array, especially with the reference sample first, and then we started using a lot of clinical isolate. And this is kind of fuzzy here, but overall the view is we could able to identify the different types of pathotype here. And then this is the green fluorescent color right at the bottom is the genes, and each character, like virulence genes, is specific for that particular pathotype. And we published this paper in Journal of Clinical. And that's it. And I believe it's time up, and it's already 1:50 here, and I'm ready to take the question. Thank you very much.
Thank you, Dr. Raghavan, sir, for a very informative talk. The question and answer session now, sir. So the first question is it possible to develop mutated region in the gene of the target virus?
Say it again one more time.
So is it possible to develop a mutated region in the gene of the target virus?
Yeah, we can do that. I mean, now, is the question is targeted towards what we can do is we can make them less virulent to make it as a vaccine. But I don't know whether we can able to induce it to change it.
Okay, sir. So the second question is, what is the pitfall in developing the vaccine against COVID as compared to influenza virus?
I'm not an expert in COVID, but what I can tell you is both of them, they fall in the same family, right? So the COVID is what right now they are targeting is they have a protein called a spike protein. So they are trying to develop a monoclonal antibody against those spike protein, because those spike protein are the one that interacts with the host cells. So our host cells, we have a receptor called as ACE2 receptor. Those spike proteins that will go and bind on to those ACE2 receptor. So people are targeting developing a monoclonal antibodies against those spike protein, and then they are just using it for treatment so that they won't allow the virus to bind on to our human receptors. So those are the thing that we can use. As far as the vaccine, I think people have been using. If you look into news, Moderna Therapeutics, I believe they have been working on using mRNA. You can synthesize RNA, and then they are using it as a vaccine. And then people are also trying to use the spike protein for the vaccine trials, and it's all in the trials right now. So you can't judge whether it will be available right now, but it's all in the process now.
Okay, sir. So the third question is, for soluble proteins, you are showing the different good vectors. But what about the membrane protein expression? Are there any vectors which can express protein into the membranes?
Yes, you can do that. So there are certain, remember, like with the protein expression is like, you can, depending upon the type of signal sequences that you use, you can dictate the protein to fall in the destination of wherever you want to put it, right? So there is a protein called as autotransporter. You can use those autotransporter signal sequences along with the protein of interest. In fact, there is one available for Salmonella typhimurium, where you can use that particular signal sequences along with the protein of your interest, and then it will allow to put your protein on the surface of the bacteria. So it is available. And remember, it's all depends on, all of them are chance phenomena, right? Sometimes, if your protein is big, it may not even express at all. Sometimes, if the protein is small, it will express, and it will express on the surface of the protein. There are a couple of good papers where they said, like, auto transporter can able to put those kind of protein of your interest onto the surface of the bacteria, and then you can use it for vaccine. In fact, in our lab, we did that Salmonella typhimurium. It's a non-pathogenic one we have isolated, and then we try to use the signal sequences, and then in fact, we tried the leak protein. It didn't work out very well, but there are other proteins where we found that we could able to express right onto the membrane.
Okay, sir. So the next question is, how can cross-reactivity be reduced in ELISA?
Yes, that's a very good question. So what you can do is, the cross-reactivity is driven by several factors, right? So the one thing that you need to consider is, it depends on where it comes from, right? So if you are expressing a recombinant protein from an E. coli, so the E. coli proteins, which you won't see in the gel, but it may cause cross-reactivity. So what we usually do is, if it is derived from the E. coli, we will always use E. coli lysate as a kind of diluent. So when you are having a PBS, when you take the serum sample, you dilute the serum sample with the PBS, right? So when you are doing that, you can take your lysate of E. coli and add those lysate along with this. In that case, you can reduce the cross-reactivity in an ELISA assay. So similarly, you can use the mammalian cells also. And it also depends on how it sits on the plate, right? The cross-reactivity, sometimes it also depends on how it sits on the plate. So you have to play around with those diluent, and also with the detergents, and also washes. Sometimes people use 3 wash, sometimes you can go for a 5 wash, 5 times to wash on the plates. So you can do all sorts of parameters to remove the cross-reactivity.
Okay, sir. So what type of screening library is used for leptospirosis antibody detection?
So the one that I used is the cDNA, genomic library. And I think most of the people, I think they did the genomic library. If you look into the literature, most of them, they did the genomic library. There are a couple of papers, they tried the phage display library, but I don't know how successful it was. And you can also do, now, the genomic sequences is pretty easy to do it. You can just take the genome and then sequence it, and then you can try to find out what are those membrane proteins in the leptospira. In fact, we did that when we had a collaboration with one of the professor at Oklahoma, and we found there are almost like 110 outer membrane proteins in the leptospiras. And then, in fact, we expressed like almost 50 or 60 membrane proteins. In fact, I published one paper where we expressed 20 different outer membrane protein. So you can do that. You can express the protein, and then you can try to evaluate for the vaccine, or you can do it for diagnostics. So it depends on the purpose. You can do the library is based on what you want to do. If you don't have the, so I told you before, right? So if you don't have the any genomic sequences data available in the database, and if it is a new disease, a new bacterial disease or a parasitic disease, the best way would be to fish out your gene of your interest is by doing a cDNA library, or you can go for some proteomics work.
Okay, so we have one last question. What is L32 or L36 of Lig-A protein? Are they leucine mutants?
Okay, yeah, I that's a good question. Sorry, no, I didn't have enough time to say about this. So LIP-L32 is what they called as a major outer membrane protein of leptospira. A group called as from UCLA, they identified this protein, and they labeled this protein as a LIP-L32. In the sense, it is lipoprotein, and it is a 32 kilodalton protein, and it's a surface protein. They found this, and then they published this protein. This is one of the major protein in leptospira. So whenever you take the leptospira and run it in a leptospira, and then take the protein and run it in a gel, you will see a major chunk at the 32 kilodalton size. So that is the LipL32. And then the next protein is LipL36. It is a 36 kilodalton protein. The same group, they identify this protein. And so what happened to this protein is, this protein, when the temperature, when you increase the temperature, these proteins get down-regulated. So you won't see this protein in infected hamster cells. But when you culture this leptospires in vitro, you will see this expression of protein. So this protein, once when it has switched over to the infected, and once when it switched over to animal, this protein, it down-regulates. So it varies, right? Some protein, it gets upregulated, some protein, it gets down-regulated. In Lyme disease, it is the same thing, where like OSPA or OSPC, some protein, it expresses only in ticks. It won't express in a culture. But once when it gets into the human, a lot of proteins gets upregulated. So depending upon the host environment, it gets triggered and it expresses the protein.
Okay, sir. Thank you, sir. Thank you, sir. Hand over the session to Dr. Agastian, sir, to propose a vote of thanks.
Okay, thank you.
Dr. Raghavan.
Yeah, yes. Yes, Agastian.
Raghavan, thank you very much for elaborative, illustrative explanations, on the discovery to diagnosis, the journey. You explained a lot. Start with how the school student, should learn not only the science but also to learn the skill. It was highly impressive, and also you have given overall view on how the recombinant protein production takes place. And you compared how the E. coli is useful and compared with the mammalian cells. And you also compared with the traditional approach and modern approach with the cloning and sequencing methods. And you also compared the linear and combinatorial approach to study the expression proteins. Thank you very much. And you have given an elaborate information on how the protein purification is done, and particularly the antigen antibody assay with with the different ELISA techniques. And and finally, you have given the roadmap on diagnosis of leptospirosis with your own research examples. And many people really understood how the genes are useful using the diagnosis by way of microarray techniques. And we also emphasized bioinformatics is an important tool. Definitely more. It will be more useful for many number of researchers. Thank you very much, Raghavan. Definitely, all the participants will take up this diagnostic as a research in the future. Definitely, the science will get momentum using this diagnostic research. Thank you very much for sharing.
Thank you very much. Thanks for providing me the opportunity to share my experiences with you all. And I believe I may not have a lot of time to explain about some of the questions they ask. If someone has more questions, they can reach out to my email. You can send me an email and see whatever that I can able to help you to figure out the problem that you guys have, and I can do that. And I greatly appreciate your time. And at this time, I thank the opportunities that the organizing committee have provided, to present some of my experiences. I'm pretty excited. Thank you very much.
Raghavan, if you come to India, please come to Loyola College. You are most welcome.
Sure, Senior.
We'll have elaborate discussions on this. Thank you. Thank you, Raghavan.
Sure. Remember, you may have to give me $1,000 for you may have to give me $1,000 because I am just it's a midnight here.
Thank you, Raghavan.
Okay,
Thank you, sir. Thank you, Raghavan, sir.
Thank you, everyone. Thanks a lot.
Thank you.
Thank you, Raghavan, sir.
Coming to the participants, I sincerely thank our Lord God Almighty for His abundant blessings and also make this the series of international webinar a possible one and a successful one. I also thank our Loyola College management, Reverend Father Rector, Reverend Father Secretary and Correspondent, Reverend Father Principal, and the Deputy Principal. Whenever I approach for this kind of event, they are wholeheartedly welcoming me and supported me wholeheartedly. I thank the Loyola College management. I also take opportunity to thank faculty members of Plant Pathogen Biotechnology Department, Dr. Ravindran, Professor Lebel, Professor Joel, Dr. Jai Jodi, Dr. Jacqueline Chinnarani, Dr. Karthi helped me a lot while from the point where I started planning for this kind of webinar seminar. I sincerely thank all the 4 speakers, starting with Dr. Saravanan, Dr. Prabhakaran, Dr. Soravel, Dr. Raghavan. They wholeheartedly accepted my invitation and delivered. Though it is difficult for them, that the timing, they accepted and wholeheartedly they did the job. I also thank the participants, who are actively participate in the question and answer sessions and also given real feedback and also participated in all the 4 webinar series. Dear participants, hope you enjoyed. Kindly excuse for any difficulties present. Your e-certificate will be given to all those who attended and submitted feedback for all the 4 sessions. And the e-certificate will be given to you in another 5 to 7 days. I also take the opportunity to thank technical assistant, Professor Samson from the Media Arts Department, Mr. Sudhagar, for their constant help and support. I also thank my research team, particularly Jansi, Amir, and Anu. I thank each and every one of you. Without your support, definitely this webinar series will not be a possible one. I thank each and every one of you. Thank you. Thank you. So with this, we close this session. Thank you very much.
Thank you, everyone.
Sir, Agastian, sir. Thank you, sir. Congrats.